Hello students, welcome to Raaonline. Our today's chapter is cell cycle. Under the section of brief discussion and question based discussion for the 2021 NEET. The question number 1, that is G1 as G2, these are the phases are noticeable in. Answer is interphase. When we study about the cell cycle, there are two phases. One is interphase and second is M phase. One is interphase, second is M phase. These two major phases of cell cycle. Interphase is preparatory phase. That is, cell prepares itself for the division, in which all events takes place like DNA replication, RNA synthesis, protein synthesis, duplication of organelles. These are all take place in interphase stage. That's what is called preparatory phase. Then the actual division takes place in M phase. The interphase further divided into three subphases: G1 phase, S phase and G2 phase. G1 as G2 phase, followed by, if we include M, so these first three are the phases of interphase, and the second is the actual phase of the in which we see the clear division of nucleus plus cytoplasm. So all four stages comprise cell cycle. That is, these are the four stages of cell cycle and their order of arrangement also: G1, S, G2, M. G1, S and G2, M. Whereas these three stages are of M phase. Question number 2. Regarding the sequence of cycle, which one is correct? I have already mentioned in the previous question, description of previous question. The order is G1, S, G2, M. G1, S, G2, M. G1 phase, actually the S phase is a key phase. That is synthesis phase or DNA synthesis phase. This is prior to DNA synthesis phase, that's why pre-DNA synthesis phase. This is post-DNA synthesis phase and followed by the M phase. So G1 is pre-DNA synthesis phase, G2 is post-DNA synthesis phase. The actual phase in which DNA is replicated, that is S phase. Then which of the following is the shortest phase of cell cycle? First three phases, they are the part of interphase. Approximate 95% time of cell cycle is consumed. 95% time of cell cycle is consumed in the first three phases: S phase, G1 phase and G2 phase. And hardly 5% time is required for the completion of M phase. In G1 phase, time required approximate 30 to 50%. 30% to 50% time G1. Then S phase, 25 to 33% time. In G2 phase, 12 to 16% time is sufficient. So as far as longest phase, then G1 is the longest, and fourth M phase is the shortest phase. That is M phase is shortest phase. M phase has two events. One is karyokinesis, that is division of nucleus takes place. Followed by cytokinesis, cytokinesis. These are the two events which are seen in M phase. During interphase, RNA and proteins. RNA and proteins are synthesized in both G1 and G2 phase. They are synthesized in G1 phase and G2 phase. That is gap 1 and gap 2. In both the phases, DNA, RNA and proteins are synthesized. G1 phase and G2 phase. Synthesis of RNA and proteins takes place. In G1 phase, protein required for the S phase are synthesized. That is, they are involved in the replication of DNA. And even we can say nucleotides are synthesized for the DNA synthesis. Nucleotides are formed in this stage. Nucleotides are formed in G1 stage for DNA. That is for the DNA. That is, preparation of S phase is seen in G1. Whereas in G2 phase, proteins for the spindle formation. Proteins for spindle formation are synthesized in this phase. That is, we can say tubulin proteins are the major proteins. Tubulin protein are the major protein which are needed in the spindle formation. They are synthesized in the G2 phase. Then come to the checkpoint. Actually, cell cycle is a complex process. Cell cycle comprises of G1 phase, then S phase, then G2 phase, then M phase. These are the 4 phases which takes place in cyclic manner. In G1 phase, preparations for the S phase takes place. And in G1 phase, there is a R point. This is checkpoint number 1. And this checkpoint, known as restriction point, and when cell crosses this checkpoint or passes this checkpoint, cell will definitely divide. The first checkpoint is G1. Second checkpoint takes place in between G2 and M. That is because cell is entering in M phase. Then M phase comprises of 4 phases. That is prophase, number 2 metaphase, number 3 anaphase, and number 4 telophase. And there is a small checkpoint is also there between a metaphase and anaphase. For these checkpoints, cyclin and cyclin dependent kinases are required. Cyclin protein, cyclin dependent kinases are required. So these are the checkpoint. Once cell enters or crosses G1, cell will definitely divide. I mean to say, in G1 stage, decision for cell division is taken. Before this checkpoint, cell can exit. That is this G0 stage. Cell can exit and may enter from this stage also. This condition is necessary for the qualified through the G2 checkpoint. Cells should be size sufficient enough, complete the accurate DNA replication. This is right. Second one is right, because DNA replication, if it is in proper manner, then only cell can enter in the M phase. Next, the phases of cell cycle in which DNA replication takes place, that is S phase. it is already known as DNA synthesis phase. S phase is called DNA synthesis phase. DNA synthesis phase. If a cell has 2C DNA, then when it crosses S phase, the amount of DNA will 4C. Suppose cell has 2 chromosomes, each one with 1 chromatid in G1. After this stage, number of chromosomes remains same. Number of chromosomes remains same. But you will see that each chromosome is now with 2 chromatids. In G1 stage, number of chromosomes 2. After S phase, G2 stage, number of chromosomes is again 2. But there is a difference in the chromosome. That is, in G1 stage, chromosomes are in monad state. That is, they have only 1 chromatid. Whereas after the passing or crossing GS phase, they have 2 chromatids in each chromosome. They have 2 chromatids in each chromosome, and the amount of DNA is doubled. The number of chromosome remains same. The physical number of chromosome remains same, but the amount of DNA is doubled in S phase. The amount of DNA is doubled in S phase. In G1 phase, RNA and proteins are synthesized. RNA and proteins are synthesized. And few cell organelles, most of the cell organelles are doubled in G1 phase, whereas in G2 phase, RNA and proteins are also synthesized, plus few cell organelles. Few cell organelles are doubled in this particular phase. So, cell organelles mostly duplicates in G1 and few in G2, few in G2 phase. So, S phase is DNA synthesis phase. S phase is DNA synthesis phase. Chromosome duplication. When DNA duplicates, chromosome duplicates. When DNA duplicates, then only chromosome duplication takes place. So, my answer is S phase. My answer is S phase. But the physical number doesn't change. Physical number does not change. Suppose at G1, a chromosome is with 1 chromatid. Then after the S phase, each chromosomes are with 2 chromatids. This is the only difference. Chromosome with 1 chromatid, here after S phase, chromosome is with 2 chromatids. This is the only difference. Number remains same. Suppose at this stage 4 chromosomes are there, then in G2 phase again 4 chromosomes are there. That is, number of chromosomes remain same, but each chromosome is duplicated. Each chromosome is duplicated. This is the question. Number of mitotic, number, that is, underline the key point. First, number is asked. Number of mitotic division required to produce 128 cell from single cell. That is, initial number is 1. Division 1, 2. Division number 2, 4. Then 8, 16, 32, 64, 128. Every time cell forms 2 cells. Each cell forms 2 cells after mitosis. First time the number of cell is 1. When it divides, 2 cells are obtained. Second time 4 cells. third time, fourth time, fifth, sixth, seventh. That is total 7 times cell divide. But this is times only. 7 times cell divide. But number of division. Initial number is 1. See, so at this stage only one cell is there. Then after first division 2 cells are there. When these 2 cells divide, then 4 cells are formed. When these 4 cells are divide, then 8 cells are formed. Then count the number of division of each cell. Count the number of division of each cell and at each plane. When we start, initial number is 1. After division number 1 completed, 2 cells are there. When these 2 cells divide, then only 4 cells are obtained. That is for the formation of 4 cells. Total division took place 3. Then if 8 cells are formed, then add 4 divisions more. That is now 7 divisions for the formation of 8 cells. Now these 8 cells divide, then only 16 cells are possible. Then add this plane number also. So each time, that is number of cells at each plane is added to count the number of mitotic division. So the answer will be 127 in this manner. That is, count the number of cells up to this level and you will find the answer, that is 127. But there is a short trick also, that is final number. Final number of cell, final number of cells minus initial number. Final number of cells minus initial number. Final number is 128. Initial number is 1. Answer is direct, that is 127. This is another short trick but applicable for mitosis only. This formula is applied only in case of mitosis. Applicable for mitosis only. Now, in order to study mitosis, best part, somatic part is best. Somatic part, body part show cell division, especially those area where cell division is taking place. That is, cell division takes place in meristematic cell and meristematic cells are available in the apical area of plant. The cells which are involved in the cell division are meristematic cells. Meristematic cells are in the state of division and meristematic cells are seen in apices of plant. That is, they are found in apex. They are found in apex. Apex I mean shoot apex and root apex. Shoot apex and root apex. Here meristem is easily available. Preferable one is root apex. So take the onion root tip to study mitosis. Preferable material is root tip and mostly onion is used. Onion root tip is used to study mitosis. Period of active mitosis, that is for the M phase. This question is regarding M phase. I have already said that interphase or preparatory phase takes 95% time of cell cycle, whereas M phase takes place hardly in 5% time. Human cell divides in 24 hours, out of which 90% time, that is 22 hours are needed for the interphase and hardly 2 hours for M phase. In yeast cell, 90 minutes is required for the division of cell. Then divide it 10% time, that is only 9 to 10%. So my answer for active division is 10% time of 5%, not 10%, 5% time of cell cycle. In that case, it may be 10 minutes to few hours. 10 minutes to few hours. When mitosis occurs in, mitosis takes place in all types of cell. That is, ploidy of an organism doesn't mean. Ploidy of an organism doesn't mean. Haploid individuals also perform mitosis. Diploid individuals can also perform mitosis. Mitosis is actually somatic division or equational division. Mitosis is somatic division or equational division. Suppose this is a cell and it has 2n is equal to 4 chromosome. In mitosis, cell divides once and genome duplicates once. In mitosis, cell divides once, genome duplicates once. Then after the mitosis, 2 daughter cells are obtained. You see, the 2 daughter cells are obtained and they have the same number of chromosomes as it was in the parent cell. Mitosis just like a photocopy. That is, identical cells are formed. That is, daughter cells are very similar among themselves as well as similar to the parent cell. So in mitosis, there is one division of cell and there is a duplication of DNA only once. One duplication of DNA and one division of cell. Total 2 daughter cells are obtained, and these two daughter cells are very similar among themselves, very similar to the parent cell also. They are the photocopy or exact copy of the parent cell. So in mitosis, we can say clones are obtained. By mitosis, clones are obtained, and single member of clone is known as ramete. Single member of clone is known as ramete. So mitosis, common in body cell, that's what is called somatic cell division. Mitosis is also called somatic cell division. Mitosis is also called somatic cell division or equational cell division. Somatic or equational cell division. Somatic or equational cell division. This is the brief discussion of mitosis, and term mitosis was given by W. Fleming. W. Fleming gave the term mitosis. Bacteria is actually prokaryote. So bacterial cell does not perform mitosis, even meiosis. There is a specific division associated with the prokaryotic cell, that is amitosis. That is amitosis. In amitosis cell division, there is a direct duplication and division of genome. This is genome duplicates, see this one, then followed by distribution of genome and duplication and distribution, direct duplication followed by the distribution, then animal cell like cytokinesis takes place. Animal cell like cytokinesis takes place in amitosis. This is all about the amitosis. Direct division, genome duplicates and divide followed by cytokinesis. You won't see that is no prophase, metaphase, anaphase or telophase like stages in amitosis. 13th, in mitosis cell do not show Golgi complex, ER, nucleolus and nuclear envelope at, that is prophase stage. At the end of prophase, all these structures disappear. All these structures disappear. Nuclear membrane, nucleolus and Golgi complex in ER. They are not visible in prophase stage. They are not visible in prophase. Suppose this is a cell. This is a cell. And this is its nucleus. And within nucleus, this is chromatin. This is chromatin. In prophase stage, condensation and dehydration takes place in chromatin and chromosomes gradually appear. Chromosomes gradually appears. Chromosomes gradually appear. After the appearance of chromosome, you see now chromosomes are now very clear. Nuclear membrane, nucleolus, Golgi body, ER, they are not visible at the end of the prophase, and spindle formation starts from the prophase stage last, late prophase. From late prophase, spindle formation starts. Then process of mitosis divided into 4 phases, their correct order. Simple question, 9th class question. Start with the prophase, metaphase, anaphase, and last stage is the telophase. These are the 4 different phases of M phase. 4 different phases of M phase. First prophase, then metaphase, anaphase, and telophase. In mitosis, there is one nuclear division, genome duplication is 1, that is cell divides once. Congregation. Metaphase stage is important phase. In metaphase stage, chromosomes show equatorial orientation. Chromosomes show equatorial orientation. That is equatorial arrangement of chromosome. This is called congregation, and spindle is clearly visible. Spindle is clearly visible. This fibrous structure is a spindle. Fibrous structure is a spindle, whose formation starts from the late prophase, but it is clearly visible in metaphase stage. And metaphase stage is the best stage to study karyotype. Metaphase stage is best stage to study karyotype. That is karyotype, that is complete morphology, complete morphological information of a chromosomes in one set. Chromosomes in one set. That is their parameter, their number of centromere in each chromosome, position of centromere in each chromosome, then arm ratio, then they are arranged according to their size. In karyotype, when we study the karyotype, chromosomes should be arranged in decreasing order. They are arranged in decreasing order. So karyotype is studied best in metaphase except shape. You can get every information. And diagrammatic representation of karyotype is known as karyogram. Diagrammatic representation of karyotype is known as karyogram or ideogram. Diagrammatic representation of karyotype is called karyogram or ideogram. So, in congregation, that is, it takes place in metaphase stage, it is nothing else, it's equatorial arrangement of chromosomes. This is metaphase plate. This is metaphase plate. So, formation of metaphase plate. Metaphase plate is formed. Metaphase plate. And these are all spindle. This is a spindle structure, fibrous structure. 16th, individual chromosomes become distinct and through a light microscope. During this phase, individual chromosomes are distinguished, that is in prophase stage. In prophase stage, and though they show maximum condensation in metaphase. Maximum condensation in metaphase stage. Anaphase, they are polarized. Anaphase, they are polarized. And in telophase stage, they gradually disappear. They gradually disappear. Colchicine. Colchicine is mitotic poison. Colchicine is mitotic poison, that is inhibits mitosis. Colchicine is an alkaloid. It is an alkaloid. This alkaloid is obtained from a plant Colchicum autumnale. Colchicum autumnale, Liliaceae family. Colchicine inhibits spindle formation. Colchicine inhibits spindle formation. So it is a inhibitor of this particular stage. Colchicine induces polyploidy. It is used for the induction of polyploidy. It is used for the induction of polyploidy. Colchicine is used for the induction of polyploidy also. So this is about colchicine, mitotic poison. Congregation takes place. Congregation, that is equatorial orientation. Answer is metaphase, equatorial orientation of chromosome. This is a cell with 4 chromosomes. Suppose this is cell and it has 4 chromosomes. Each chromosome is arranged at equatorial plate. Each chromosome now arranged at equator. This is congregation. This is congregation. That is equatorial orientation. And this is a spindle. This is a spindle. Spindle is fibrous structure. Spindle is fibrous structure, and spindle formation requires a specific protein, that is tubulin protein. For the formation of a spindle, tubulin protein is needed because spindle is made up of microtubule and microtubules are made up of tubulin protein. Microtubules are made up of tubulin protein. So this fibrous structure, it is spindle-like because it is thin on both the end and thick at the center. So the shape is actually spindle shape, but when we see, it is a fibrous structure, comprises of 3 different types of fiber. Pole to pole, that is continuous fiber. Pole to chromosome, these are called chromosomal fiber or tractile fiber. And when in anaphase stage chromosomes separate, then some fibers are seen at this place. They are called interzonal fiber. Number 1, continuous fiber, that is from pole to pole. Then pole to chromosome, they are chromosomal fiber. And at the time of polarization, fibers are seen in between the 2 separating chromosomes, that is known as interzonal fiber. They are called interzonal fiber. Formation starts from late prophase, but it is clearly visible in metaphase. That is, complete spindle is seen in metaphase stage, though its formation starts from the late prophase stage. Its formation starts from the late prophase stage. And if we see the structure of chromosome, these are the two chromatids. They are joined together at centromere, and on each side proteinous plate is there, that is kinetochore, and kinetochore is site of attachment of a spindle. 19th question, that is best stage at which total number of chromosomes can be counted. I have already mentioned, except shape, all informations can be gathered from metaphase. Metaphase stage is best phase. Metaphase stage is best phase to study entire karyotype except shape. In metaphase stage, you can count the number of chromosome. Number of chromosome can be counted. Number of chromosomes can be counted. Second, their size is measured. Right? So, metaphase stage is best stage to study what? Karyotype. Karyotype, that is complete information of chromosomes of a genome. Except shape. Shape is studied in anaphase. Shape of chromosome is studied at anaphase stage. The rest of the informations can be taken from the metaphase stage. 20th question. The characteristic of stage of mitosis where there is a separation of sister chromatids. Answer is anaphase. See this one. In metaphase stage, chromosomes are at equator and with two chromatids. They held together by means of centromere, and they are in equatorial region. In anaphase stage, there is a division of centromere. So, these two chromatids which are joined together by means of centromere get separated. So, first of all, division of centromere. Then followed by separation. When centromere divides, these two chromatids of chromosome separates and each one is now known as sister chromosome. Better to say daughter chromosome. Now they are termed as daughter chromosomes. These are known as now daughter chromosomes. And when they are joined together by means of centromere, then they are called sister chromatids. Then these are the sister chromatids. But after separation, each one has the status of a chromosome. Has the status of chromosome. And this is all seen in anaphase stage. Spindle apparatus. Number of times we have discussed in the previous section also. Formed during which stage of meiosis? Formation starts from prophase but completely visible in metaphase. Formation starts from prophase but completely visible in metaphase. So depending upon formation, if formation start, then prophase, then its formation complete and visible, then metaphase stage is correct. Mitosis results in constant number of chromosomes in the daughter cell that it was in the parent cell. Suppose parent cell has 4 chromosomes and 2 genomes, then at the end of mitosis, 2 daughter cells are obtained, and they to have the same number of chromosome as it was in the parent cell. So no change in the number of chromosome, no change in the number of chromosomes.